Thank you so much, Randy, for inviting me. And I'm delighted to share some very interesting aspects about how we understand how mammals evolved their skin structure, just to fit the right theme for DinoFest for all things living in the past, but in their living colors. Mammals are incredibly diverse, and one aspect of our great diversity is in our skin structures. Most mammals got great hairs with a coat of fur, but in addition to this hair structure, we also got incredibly rich variety of skin glands, say like mammary glands, sweat glands, sebaceous glands, so on and so forth. And this hair-related structure can also display tremendous amount of diversity. For example, some mammals have spines, and the spine can have a variety of sizes, and the mammals can have all this, the keratinized scales, such as the pangolin or pinecone mammal. But the mammals can also develop osteoderm, what we call the bony scutes in the skin. Okay, so one of the great question is, so if we have all these diverse skin structures, and how we define a mammal. So let me rephrase it, and what makes a mammal from the perspective of skin structure. The first important feature that makes us mammals is all female members have functional mammary glands for lactation of the young. Because of this evolutionary innovation, our young, our fetuses grow in an entirely different mode as compared to all other non-mammalian vertebrates. But mammary glands is just one variety of skin glands that are related to the hairs. Of course, mammals are defined by our hairs. This coat of furs is very important, but it's for a very interesting reason. And our hairs are actually one of the better tactile sensory organs for our body. We have much better tactile sense than all of the non-mammalian ammonites. Of course, furs and its subcutaneous fat and the sweat gland are great structure to help us with thermal regulation. Mammals have elevated metabolism that we generically call them warm-blooded, but with this, we do have the need to keep our heat, and when the environment gets super hot, we also sweat to dissipate the heat. And this hair-related structure can reorganize itself, reshape themselves, so that we can develop scales, we can develop spines, and we can also develop scutes. So as a paleontologist, I'm absolutely interested in how in our evolutionary history, we can trace the origin of the critical, important, highly distinctive mammalian features. So the general way we approach this is we would go deeper in the fossil record. And modern mammals and its common ancestor goes back to about 165 million years into early or middle Jurassic. But this modern mammal group, with all these characteristics of spines and hairs, so on and so forth, are actually a subgroup of a more inclusive phylogenetic lineage or clade we call mammalia forms. And this mammalia forms, in turn, is a part of a larger group called synapsids. And essentially, modern mammals goes back to at least 165 million years. And the more inclusive group, mammalia forms, will go back to about 220 million years. And still yet more inclusive group called synapsid would go back to 310 million years. So this concordance of the geological sequence and our phylogenetic evolution gave us a very reliable framework to infer how any of the features would evolve in mammalian history, of course, including hairs and their integumentary related structures. So our question would become very simple, okay? We know that in therapsids and the basal cynodonts from late Permian have fossilized skin. And their skin is leathery and perhaps a little scaly. But we also know that modern mammals have furs and the furs related structure. So the question becomes very simple. We just follow this phylogenetic map and we just try to trace whatever fossil feature that can be interpreted to be hair and a hair-like structure in all these transitional forms that we call mammalia forms, okay? And so in the recent 20 years, mammal paleontologists had tremendous luck of all this very interesting fossil finds. And we have some great fossils. I'm going to discuss a little bit more from the phosphate preservation in early Cretaceous from Spain, but a very large body of evidence are really from this compression of the hair structure in all of this transitional mammal, what we call the mammalia forms from the Mesozoic of China. One of the great lucky find that we had was this particular fossil called Castorocauda. Castor stands for castor, that's the Canadian beaver. Cauda means tail. And this fossil gave us a very important indication that the fur-clad mammal can actually develop some scaly structure on its tail. So this was a big breakthrough in 2006. And then about 2013 up to 2015, we have another wide range of fossils that also are fossilized with fur. So this particular group called the Docodon mammalia forms is our near relatives. And these are one of the first indication that modern mammal like hairs, spines, scutes are actually originating long before the common ancestor of modern mammals. Okay. Another very important group that gave us the similar kind of information is called Herramiids. You know, in our version of a phylogeny, and these Herramiids are placed very basal in the cynodon to modern mammal evolution, and they are mammalia forms. And one of the first find of this particular group is called Megaconus, and it's really preserved with this very rich and dense hair all around its body. And another related form is reported in 2017. And for this, we clearly have the skin association of skin membrane association with the limbs and cover the whole body. And so we call it Maya patagium. And essentially, it's the mother of the winged mammals from about 160 million years from, again, Jurassic of China. And now we have another 3 or 4 different species of this particular group called Herramites, and they are all fossilized with this skin membrane. And we can literally tell that they have a tail section of the membrane called uropatagium. And we can also identify a plagiopatagium, that's the side wing. And then we got a stretch of skin between the forearms and the neck, and that's the forewing. So this long extinct relatives from about 170 to 160 million years ago are just like our modern mammalian gliders with all the skin structure that are terrific for flying. And this particular fluorescence light false colored photograph is a good demonstration that this hair structure is actually absolutely contiguous and cover the entire body. So mammals are flying way back in Mesozoic, and we know this because we found the associated soft tissue structures like hairs and like the skin membranes. And this figure very large with our broader understanding of mammalian evolution. From 30 years back, we had a biased perception. This biased perception was Mesozoic were always dominated by big dinosaurs, and therefore mammals are generally restricted to the small body size and generalized terrestrial adaptation. It's only after dinosaur went extinct, ecological niche become available, mammals start to develop all variety of ecological specialization, like say semi-aquatic mammals, general terrestrial mammals, and the tongue-feeding mammals with armor cover like armadillos. And we also have tree climbers. Once we move on the tree, we started gliding. But this broader perception that the Mesozoic mammals are ecologically restricted and very simple is actually from lack of evidence from way back, but we have entirely different view now, okay? So starting from 1993, 1995, we start to find the first semi-aquatic mammals, such as Castorocota. And then about 2005, paleontologists also discovered mammals can get pretty big, up to 10 to 12 kilograms. So there are dog-sized mammal that can actually feed on other small vertebrates like small dinosaurs, okay? And then in 2005, we start to discover the very first tongue-feeding mammal associated with digging adaptation, sort of like our extant armadillos, but we have not yet found the scutes back then. And then the Cretaceous mammal have a wide range of tree climbing taxon. And once you climb on the tree, it's only a short jump away to become a glider. And that was about 10 or 12 years ago. Since then, we have made even more discoveries in the Mesozoic mammals. The very first group that we did not even know was very rich are now represented by dozens of specimens, at least 20, 30 different species. And these are the Herramites, and we can go from terrestrial form all the way to the gliders. And if we add the Docodon mammalia forms to this picture, in Middle Jurassic, the mammalia forms is already developing the same range of ecological diversity as the small modern mammals. Isn't that amazing? If we add onto the Cretaceous form, we get incredibly enriched picture about how diverse, how varied, and how disparate our distant mammalian form relative actually are. So this well-preserved skin structure gave us that very important hint to reinterpret our broader view about early mammal history. Now I'm going to switch the gear to talk about how we can understand even better beyond the fact that there is a halo of hair around the body. What about the hair structures? Okay. And we all understand extant mammals are very specialized, diverse in their own right. We can also have spines, and we can also have scutes, and we also can have osteoderm, and these are all very distinctive and disparate structures. And our big break comes foremost with Castorocauda, and this is the first mammal going back all the way to 160 million years with preserved scales. That by itself is a big deal, okay? And so here is a good picture about how we look at the local area besides the tail, and that gave us this capacity to reconstruct a broad scaly tail. The fact that got the broad scaly tail plus fish-eating teeth, we can reliably interpret that this is the very earliest known semi-aquatic mammals. And about 10 years ago, 2012, Dr. Angela Buscanione's team discovered an incredibly well-preserved mammal. Later, we named this as Spinolistis xenopterosus. And this is from Las Joyas quarry of Barremian age, about 125 million years. And this particular fossil site got one incredible preservation condition, and that is all of the vertebrates have been permineralized with the skin tissue or skin-related tissue. The general understanding is the particular site really facilitate phosphatization very well. And with that, we have this incredibly interesting fossil. Okay, and here are one part of the fossil. Here is another part of the fossil. One part of the fossil to facilitate research is transferred into a plastic cask so we can look at the fossil from both sides. And a key feature about mammalian skin structure is what we call the compound follicle. Let me explain this a little bit. Most of the mammals have a simple hair. Most simple hair structures in mammals have a single follicle and it'll grow a single primary hair. But another entire different class of hair structure always grows secondary hair. It's clustered together. And down inside the skin, we have a compound follicle. Okay. This compound follicle is of fundamental importance. In addition to the follicles, of course, we would have skin glands, we have sebaceous glands, we have skin muscles, so on and so forth. In the early development, if this compound follicle gets reorganized, it would turn multiple bundles of hair into spine, or it will turn multiple bundles of hairs into a cluster of spine. And that's exactly what we discovered in the phosphatized skin of Spinolistis. Here are some structures. So here is a hair bulb, and here is a hair bulb, but this hair bulb from gross morphology clearly supported one primary hair and plus a whole bunch of secondary hairs. And here is a scanning electron microscope scan of the surface. Here is the orifice of the follicle. Here is the primary hair. Here is second hair. And when this hair start to grow, it exactly have the same kind of cuticular structure, scaly pieces that stack up in a column fashion that makes up all the diversity of mammalian hair in different arrangements. So here we really got it nailed that starting with spinal listis, which is a triconodon, we start to have the first modern mammal-like compound follicle, which is the foundational structure for modern mammal to develop spine. We do not have a very long spine in the spinal listis fossil, but nonetheless, the spine is there, and especially on the dorsal side of the animal, the spine is about 5 to 6 times thicker than the thickest hair. And in addition to that, in a scattered manner along the back of the mammal, we also have this dermal scutes. Okay, and what's even more interesting, but unrelated to the skin structure, is this phosphatization is so thorough, and we think we can recognize an area of rich concentration of iron, therefore it's red. We interpret it to be the liver, and whereas the front portion of it, the texture of the preserved permineralized soft tissue is very similar to the lung. And the boundary of this conform to the diaphragm, which is the breathing muscle to separate the lung region from the abdominal cavity, more or less according to its position in all of the modern mammals. So what we have to tell, essentially, we are always interested in finding out how the diverse mammalian skin feature that makes us so diverse, so appealing, so fuzzy, and so lovable, how do we trace them back into the Mesozoic history? With these findings of a spectacular range of mammalian fossils, we can, in some cases, reconstruct with confidence that our hair-like structure of extinct mammals can be traced long way back, at least to the late Triassic. So with that, if the audio works well, I will be happy to discuss and entertain your questions.